Okay, so at this moment, we are going to go through DNA history, and I hope that you listen to the words that are about to come out of my mouth. This PowerPoint over DNA history is going to end up being very important for lecture exam 4. So you're going to want to know who discovered what. Okay? Okay? Oh, I can't use that symbol anymore, can I? White supremacists. Okay, yes, we're going to really pay attention to this PowerPoint. That's what we're going to do because it's super, super important for your lecture exam 4. I'm going to find the PowerPoint eventually, and I'm going to start the PowerPoint with the right slide. There we go. I don't know if you know this, possibly you do, possibly you don't. Historically, DNA was not known to be the genetic material. It wasn't even hypothesized to be the genetic material. Human beings assumed that proteins were the genetic material, and there's very numerous reasons for them to have thought that. One, you are 80% protein by dry weight. It's the most of what you are besides water. Two, proteins do everything: hair color, eye color, facial structure, body structure, size, weight, all of it is determined by proteins. Three, proteins have a much larger language that they speak in than DNA. DNA only has the 4 base pairs of adenine, thymine, cytosine, guanine. And so in terms of just looking at quantitative data, you would assume, possibly back 200 years ago, that proteins beat DNA due to how they were manifested in the body. But we do now know that DNA is the genetic material. How did we get there? Whew. The march to understand that DNA is the genetic material was followed by these people: T.H. Morgan in 1908, Frederick Griffith in 1928. He's going to be an important one. Griffith is Avery McCarty and McLeod, 1944. Erwin Chargaff in 1947, also an important one for you. Hershey and Chase, super important. Theirs is the classical experiment when you talk about DNA discovery. And then, of course, obviously, Mr. Watson and Mr. Crick in 1953. Since then, Mendelssohn and Stahl have clarified some of the questions we had about DNA. T.H. Morgan, while not important to your exam, found out that chromosomes relate to phenotype. So phenotype, the definition of phenotype is the physical manifestation of a gene. And then chromosome is going to be, you're talking about the gene itself. And so you typically talk about genotype and phenotype. Genotype is the DNA strand itself, and then phenotype is how it physically manifests after it's been copied and translated and transcribed into protein. So T.H. Morgan worked with a species known as Drosophila. It's your fruit fly. It's your classic fruit fly. It's a species that we still work with massively to this day. And the reason for that is they breed so quickly that you can see what multiple generations are going to do in about 10 days. And it really gives you fast results when you manipulate genes. Excuse me, when you manipulate, when you manipulate genes. So he associated a phenotype with a specific chromosome. And what he did is he found out that white-eyed males here, because your wild type is typically red eyes. So red eyes is normal, what you would find in the wild. White eyes is going to be a mutation. They had a specific X chromosome. Oh, and so this is known as a chromiograph. It has other words as well. But you can see that they have isolated each individual chromosome here. Okay, so genes and chromosomes. Well, okay, his conclusions are that genes are on chromosomes, okay? So chromosomes are just lots of genes, and each gene codes for a different protein. But they did not know at this time in 1908 if it was the protein or the DNA of the chromosomes that were the gene. We initially taught protein, told you all the reasons why. I want you to look at this picture, please, very closely. So here is condensed chromatin. Now, when chromosome is condensed, it's not workable. We can't transcribe and translate it. We only condense our chromosomes in order to go through mitosis, and then we relax the chromatin into what we call a chromosome, so we can work on it. In order to be able to condense our chromosomes, we wrap them very, very, very tightly using methyl, which you can't see, but methyl. Methyl is positive and phosphate's negative, so they're opposite charges, so they like each other. You use methyl groups to wrap the DNA, the chromosome, around what we call histones, and histones are balls of protein. So here's a histone with chromatin wrapped around it very tightly in order to form this condensed chromatin here. When it's all the way relaxed, when we have it unwrapped from the histones, we can work on it. And this is when we do our transcribing and translating and replicating as well of the DNA when it's in its completely relaxed state. So that's as far as Thomas, what was his name? Morgan got, T. Morgan. That's as far as he got in terms of, we found out genes are on chromosomes. We still don't know if it's the DNA or the protein that's the important part. Frederick Griffith, please pay attention to this. He figured out the transforming principle. This has to do with bacteria. Bacteria have multiple ways to transfer DNA between themselves that have nothing to do with their chromosome. They have things called plasmids. And what plasmids are very, very tiny circles of chromosome, chromosome, but not the full chromosome in the nucleoid region. They're like separate little circles. They usually code for one gene. These little plasmids can jump from bacteria to bacteria in different ways. They can be transduced, which is when they're carried by a bacteriophage. Don't worry about that. They can be transformed, which is where live bacteria pick up these plasmids after they've been released into the environment from dead bacteria. Transform, transduce, and trans something. There's a third way, and then there's actually bacterial sex, where they form a conjugation bridge between two bacteria and can physically push the plasmids from one bacteria to another. So there's 4 ways bacteria exchange DNA. I can't remember one of them, apologies. But transforming is where live bacteria pick up these plasmids from dead bacteria after these plasmids have been released into the environment from the bacteria's decaying. So Frederick Griffith, here with some sort of terrier, worked with Streptococcus pneumoniae bacteria, so the common cause of pneumonia. He was working to find a cure for pneumonia. He had 2 strands. He had this harmless strand of Streptococcus pneumonia that did not have the potential to cause pneumonia. It wasn't pathogenic, and it was rough, which means that its coating on the outside had texture to it. And then he had smooth, pathogenic Streptococcus pneumoniae that he heat killed, because remember, they also only live in viable temperature ranges, bacteria do, and you can just boil them and kill them. So these pathogenic bacteria caused fatal disease in mice. So here's a smooth capsule. You see that, how smooth it is here. He would inject it into mice, they'd get pneumonia and die. There he is, sorry. When he would heat kill these smooth pathogenic bacteria, all of a sudden, the harmless live bacteria would turn into smooth pathogenic bacteria. That's interesting. So he found a substance that passed from the dead bacteria to the live bacteria and changed their phenotype. This is called the transforming principle. So here, smooth. Smooth is pathogenic, it's deadly, the mice die. The rough type, which is just shown here without a capsule, not pathogenic, they don't kill the mice. Heat killed pathogenic doesn't kill the mice because it's killed. You can't kill mice if you yourself are dead. But when he mixed the heat killed pathogenic with the rough non-pathogenic wild type, this is dead bacteria This is live bacteria. Somehow the live bacteria transferred into pathogenic bacteria and the mice died. Oh, and here's all the words of what I just said. Okay, something was happening that was transferring pathogenicity from dead bacteria to live bacteria, and we call that transformation. Now, he called it transformation because he saw a transform in the phenotype. It went from rough and non-pathogenic to smooth and pathogenic. We call it transformation based off of that, but what's really happening is the rough wild type bacteria are picking up in vesicles the plasmids from the dead heat-killed pathogenic bacteria. Griffith is very important for your exam. Avery, McCarthy, and McLeod, not important for your exam, but they found out that it was DNA specifically that was being picked up by the pathogenic, by the non-pathogenic bacteria. They purified both the DNA and the proteins separately. You can separate DNA and proteins by weight. Proteins are lighter, I think. Don't quote me on that, but I think. And they separated them. And so they had a solution of only the proteins and a solution of only the DNA from those heat-killed pathogenic bacteria. And they injected the protein into, or they mixed the protein in solution with the wild type and injected that into mice. Nothing happened. The mice were fine. They injected, or they mixed, I'm sorry, the DNA part with wild type protein, with wild type non-pathogenic bacteria. The wild type bacteria turned pathogenic and killed the mice. And so they were able to say, hey, it's the DNA. The DNA is doing it. And so this was the first experimental evidence that DNA was the genetic material. That's huge. It should probably be on your test, but whatever. Avery, McCarty, and McLeod. Don't they look like fun party people? Now, Hershey and Chase confirm that DNA is the genetic material. Their study is classic. Hershey and Chase are always pointed to as this is how you do experimental science. This is a study that you will study in research, in biology classes and whatever, because it's simple and it's beautiful in its simplicity. I mentioned bacteriophages before. Here's what a bacteriophage looks like. It's a capsid, which you're going to learn about viruses over Thanksgiving. And a capsid is a protein shell with DNA or RNA, so genetic material inside it. In this case, it's a DNA. Bacteriophages themselves go on to have this protein coil and these legs or tail fibers, and so they look kind of like an alien. Most viruses stop at just the capsid with the genetic material inside. Here is an electron scanning micrograph of bacteriophages. We do actually know this is what they look like. This has been falsely colored for you to show DNA being injected from the bacteriophage into a bacterial cell. So phages, bacteriophages, are viruses of bacteria. So Hershey and Chase, it's a classic blender experiment, and not joking, they used a blender, but they worked with bacteriophages. And they grew 2 phages in 2 media. Now, at this point in time, we had found out the general makeup of DNA and proteins. Right? So sulfur is found in proteins. Remember, we had those sulfur or disulfide bridges I was showing you with the thing about how hair gets permed curly because you make the sulfur bridges, and that's why hair salons used to always smell like rotten eggs. Sulfur is in proteins, and it holds that tertiary and sometimes quaternary structure together. Remember, disulfide bridges are pretty much the strongest bond we can make in biology. Sulfur is in proteins, but what could be in DNA that is not in proteins? A lot. That is not a lot in proteins, we'll say it that way. But definitely is in every nucleotide of DNA that we can radioactively label phosphate. Sulfur is in proteins, phosphate is in DNA. So they grew these bacteriophages, which what that really means is that they had a petri dish full of bacteria that the media that the bacteria were feeding on was radioactively labeled with either sulfa 35 or phosphate 32. And then they put bacteriophages in it. And so the bacteriophage is going to go and infect a bacteria, take over its machinery, make its own DNA and proteins, package them up, and as they leave the bacteria, they kill the bacteria itself. But that means that the DNA and the protein that has now created that bacteriophage is radioactively labeled with either, well, the proteins with sulfur 35 and the phosphate with sulfur 32. We talked about why sulfur and phosphate. So here we go. We send bacteriophages in. Here is sulfur in the protein shell. Here is phosphate in the DNA. The bacteriophages will infect bacterial cells. So these are the bacteriophages we grew in that radioactive media. Now we're infecting non-radioactively labeled bacteria. Okay, you with me? You see here that this protein stays on the outside of the bacteria while the DNA is injected inside. And after a little bit more study, it wasn't as simple as noticing this. They found out that the sulfur was found in the media, but the phosphate was found inside the bacterial cell. Let's go a little bit farther. Okay, so which radioactive marker is found inside the cell? We can see that it's the phosphate. It gets injected inside. Which molecule carries viral genetic info? Once again, it's the DNA. In order to separate by weight the sulfur proteins from the phosphate DNA, they mix it in a blender. 1952. This is their high tech. It's a blender. So they mixed radioactively labeled phages with that bacteria. They infect the bacteria. They agitated it in a blender to separate the phages, the sulfur protein shells that were sitting outside the bacteria, those just shook off and were floating in the media, from the DNA inside the bacteria. Bacteria is heavier than viruses. So by blending it, the bacteria would sink to the bottom. Well, okay, so they blended it and then they had to centrifuge it because a blender is just not fast enough. I don't know if you've ever seen a centrifuge, but it is a machine that is enclosed because you are spinning at high revolutions to separate things by weight. So the bacteria is going to fall to the bottom while those capsids, the viruses, the bacteriophage pieces will stay in the media. Okay, so they found out that the proteins stayed in the media and the phosphorus was found with the bacteria. Whoops. Therefore, viral protein does not enter bacteria. So viral protein can't be responsible for creating more protein by hijacking bacteria machinery and having it make its own DNA and proteins. It's not the proteins. They found out it was the DNA. The DNA is what enters the bacteria. That's the transforming factor. Here is Chase and Hershey, Hershey and Chase. You always, the male always came first back then, Hershey and Chase. And then we have Erwin Chargaff, and he is very important. Hershey and Chase is very important. Griffith is very important for your exam. Chargaff came up with rules about DNA. He spent his whole life on it. Here he was young, here he is old, doing DNA. And he came up with two very important rules, wink, wink. One, that DNA, the nucleotides, the base sequence, varies from species to species. Two, this isn't two, but not all four bases are an equal quantity. Bases represent a ratio. What is the ratio? Sorry. The ratio is that A's equal T's and G's equal C's. The ratio is that adenines equal thymines and guanines equal cytosines. And yes, I realize that the point system is off here, but roughly A's equal T's and G's equal C's. So varies from species to species, A equals T, G equals C. Those are Chargaff's rules. And he was right. And then of course we have the structure of DNA by Watson and Crick. In the end, it wasn't just Watson and Crick. You got to understand, everybody had the same amount of information in the biomedical world at the same time. Hershey and Chase figured things out. Chargaff figured things out. Morgan had figured things out. Griffith had figured things out. Everybody across the world who was interested in working on this had all the same information. They were all just frantically trying to figure out how it fit together. Watson and Crick historically will always be noted as the two gentlemen who developed the double helix model of DNA. What they don't say is they didn't do it on their own. They say that now because now we know. At the time, they were just lauded, which they were praised for being the ones. But you had other leading scientists working on the same exact question. You had Linus Pauling, you had Rosalind Franklin, and I'm sorry, I don't know Wilkins' first name. Maurice, I do. And you had Maurice Wilkins, all working on it. And that wasn't all. Those were just the leading men. Oh, look at that. I was right. No, I'm so proud of myself. Yes. Watson and Crick actually had it wrong originally. They had the DNA bases outside, pointing outside. Adenine was over here and thymine was over here, and they had the sugar phosphate backbone down the middle. They could not figure it out. They couldn't figure it out. They had no idea what they were doing wrong. Then they saw this. This is an X-ray crystallography that Rosalind Franklin actually took. They stole it from her desk. No joke, straight up stole this from her. But what this is showing you, this perfect X pattern on the inside of this X-ray is showing you that the bases are inside. And that's all they needed. The second they had that information, they knew that A is equal T's and G's equal C's, so they have to go together, A to T, G to C. They knew that it was a sugar phosphate backbone. They knew all the other information. They just couldn't figure out where the bases were. Boom, they're in the middle. They wrote a one-page paper, published in Nature. First, they were the first to publish, which means history will always say that they were the ones to discover it. Whatever. Now that we had discovered it, I've done this before. This in your replication PowerPoint, the one where I filmed myself going through the PowerPoint, the line from an article in Nature is something along, it has not escaped our notice that the structure of DNA implies a mechanism for copying, is the very last sentence of their paper. It's not those words exactly, but it's basically that. In terms of science, that is what's known as a mic drop, because that was the next big thing. All right, we found out DNA. We now know DNA structure. How does DNA pass from one to the other? So that's going to be the next big step, is how does replication occur? And for these two to finish their paper with, like, it seems like there's a very obvious mechanism for replication to occur. Mic drop. Like, you know, that's not what you think of a mic drop, but it is. Crick, I believe Crick, I'm pretty sure this is Crick. One of them is still alive. One of them passed away already. Rosalind, unfortunately, for a long, long time, never got credit for anything. Only posthumously has she ever been credited with her role in the discovery of the DNA double helix. She died from cancer due to X-ray exposure, because she was doing X-ray crystallography. That was her job. So she died from cancer at a very young age. Okay, so then the next question is, how is DNA copied? Knowing that A is equal T's and G is equal C's, and knowing that it's a double helix with the bases to the inside, the base pairing suggests that it will allow each side to serve as a template for a new strand. Now, we went through a lot of more science to figure that out. But it turned out, Oh, here it is again. I'm always crazy. Here's the line. I'm sorry. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. It might take you 20 extra years to figure out how beautiful of a sentence that is, because I've never appreciated it until I was in my 30s. But it's a beautiful sentence. What happens is we found out, yes, here we go, that, well, no, what science had to find out was how was DNA copied. We had many different models. We had completely conservative, where you had the two parent templates stay together and the two daughter strands create the new genetic material. But if you see here, down here in the second generation, that doesn't really pan out because you don't have equal amounts of anything. Semi-conservative, a parent strand serves as a template for a daughter strand. And so each template is old and new, or each DNA is actually old and new. And then dispersive, were like bits and pieces were copied and old, and bits and pieces were new, and that doesn't work either. So these all became experimental predictions. Can you design a nifty experiment to verify? Sorry. What this is showing you, stop it, is this is the parent generation. This is the F1 generation, which is important when we get to genetics. And this is the F2 generation. So familial 1 and familial 2. Anyway. Then we get Mendelssohn and Stahl. A few years after the discovery of the DNA double helix structure, and what has been called the most beautiful experiment in biology, if you will, confirm that replication is done semi-conservatively. And once again, we had to label things radioactively. Radioactive. So they labeled parent nucleotides with a very heavy nitrogen, N15. So they created cells in an environment, in a media, where the only nitrogen available was N15, and we know that adenine, thymine, guanine, and cytosine are very heavy in nitrogen. They're nitrogenous rings is what they are. So of course the nitrogen has to be incorporated into the DNA if we're going to make DNA. Then they had media containing a lighter isotope. They transferred parent DNA into this new media, so DNA that definitely had the N15 incorporated. They then put into the new media and made sure that environmental conditions were absolutely right so that they would replicate and or undergo mitosis and create daughter cells, right? When that happened, they killed everybody and got the DNA out of it and then centrifuge, once again at a really high rate. Now we have two different size isotopes. We have a heavy isotope that's going to be at the bottom of the centrifuge, and we have a lighter isotope that's going to be higher up in the centrifuge. So what are your predictions? Anybody? Anybody? Was everything heavy? Was everything light? Was it a mix of both? And so it was completely even in the middle? So that would be dispersive. So that would be dispersive. If it was completely heavy, everything would be at the bottom. If it was heavy and light, sorry, once again, if it was completely heavy, everything would be at the bottom, and that shows conservative replication. If it was heavy and light, there's going to be two bands in the centrifuge of the heavy parent DNA and the lighter daughter DNA. And if it was a mix like dispersive, then they would have to have all in the same band because equally it would weigh out the same. What's your prediction? We now know it's semi-conservative replication. It kind of ruins your prediction. So, okay, let's just get it all up here. So the first round of replication, this is what you I'm so sorry. This is what you expect to see. Your parent strand would be at the bottom. Apparently, no, I was right, but I didn't think, okay, doesn't matter. Oh, no, that makes sense. So your parent strand and conservative, let's go back to conservative. In conservative, you would have the parent strand completely of old DNA and the daughter strand of complete new DNA. In semi-conservative, you have one of each. And then in dispersive, they'd have to equal out pretty much the same, right? Okay, so, no, I'm going the wrong way. Okay, so I was slightly wrong in how I was explaining things, so that's why there's pictures, right? Can't always trust me. In the first generation, okay, this is what you would expect in conservative. Here's what you would expect in semi-conservative and dispersive. In the second round of replication, when they actually had the parent strands transferred to the new media, this is what they would expect to see if it was conservative. If this was semi-conservative, they would expect to see the two bands of different weights. And then dispersive would have to only be one band. And what did they end up seeing? So that shows that that doesn't work. I don't know what the mouse symbol means. But in the F1 generation, we only see one band. So what they're saying is that it's absolutely not conservative. So we have to figure out if it's semi-conservative or disruptive, because we have the one band there. And then we see in the second generation that we have two bands, which means that semi-conservative is the only way that it can be. I don't think it's the most beautiful experiment in biology because it still confuses me a little bit, but that's fine. Mendelssohn-Stahl, more party people. Look at them party. They are partiers. Look, this is at Harvard University and they're just drinking it up. So the march to understand the genetic material, I told you followed these people. This is a very important slide for you historically. Morgan in 1908, genes are on chromosomes. Griffith, a transforming factor can change the phenotype. Griffith couldn't figure out if it was proteins or DNA. He just knew that something's changing the phenotypes. Avery, McCarty, McLeod, it's DNA. Chargaff, hey, DNA follows some very specific rules. It's different in every species, the base composition, and A equals T and G equals C. Hershey and Chase, confirming all that history, that yes, DNA is the genetic material with that classic blender experiment with the bacteriophages and bacteria. Watson and Crick stealing other people's work to determine the double helix structure of DNA. Mendelssohn and Stahl answering the question of how does replication occur. Then you had to have all other scientists figure out how does DNA turn to RNA, turn to protein. This is the central dogma of biology. If biology was a religion, this is the golden rule. This is it. This is like saying the Holy Trinity in a Catholic church, or this is like saying Allah is the only prophet in Ooh, I hope I didn't get that wrong. Okay, if I did, I do apologize. But DNA replicates, it's self-replicating, to RNA through transcription, to protein through translation. Nothing that we have found doesn't follow this rule. This is the basis for evolution. This is the basis for living, that DNA is transcribed into RNA and is translated into protein. And so that's what we are learning all this week. How does DNA self-replicate? How is it transcribed into RNA? And how is it translated into protein? There have obviously been many other scientists after Mendelssohn and Stahl to figure out these steps. But this is the central dogma. This is the rule. This is it. If there was, how does biology work? DNA to RNA to protein. The whole 1,000-page textbook you have is based on DNA to RNA to protein. So obviously you can be a scientist and still have some fun. If you have questions, you know how to find me. Thank you so much. Let me stop share and then let me pause. Bye now.